Hi, everybody. So this is the second of my 2 recorded lectures for you for after my surgery. So this would be typically our Thursday, the 3rd of March. So we were in the middle of chapter 6 on viruses and prions, but I'm going to start this class as I usually do by putting up our whiteboard with our homework for the week. I was going to warn you, I am recording this on Saturday evening, and my husband is watching hockey, and being married to a French Canadian, we actually have a hockey goal siren in our living room. So if his team scores tonight, you might hear the siren in the background. So please disregard unless you're a Montreal Canadiens fan, and then you can cheer or something. Anyway, for homework this week, again, we have our chapter 7 study module, right? That's due on the following Monday. And then we have our Sarago check-in number 2. The levels for that are for chapters 1 and 2. You should be at a 3.0 for chapters 3 and 4. You should be at a 2.5. Chapter 5, we're going to do that at a 2.0. Chapter 6 at a 1.5. And last but not least is our chapter 7. We should be at a 1.0. Remember, though, this has 2 sets of vocabulary words. Both of these, the chapter 7 study module and the Sarago check-in, are due Monday, and that is the 7th of March. The other thing that I did want to point out, which I pointed out in my last recorded review, is that we have exam 2, which will cover chapters 5, 6, and 7. Remember, it will be a little bit 5 heavy because that's where we spent most of our time. So there will likely be close to 20 questions for that, and maybe 15 and 15 for chapter 6 and 7. I'm not sure the exact breakdown, but it's something close to that. That will open at 5 p.m. and that would be Thursday, the 10th of March. And then this will close at 5 p.m. Saturday, which is the 12th of March. Now, we should see each other long before this, right? We should have class on both the 8th and the 10th, where I should be back, unless there's some unforeseen complication, but I imagine that I will be back on the 8th. But that is all the homework and kind of the exam. Please note that there is a review outline in Blackboard for this exam too, so you're welcome to start working through that if you have not already. If you have any questions about the study module, the Sarago, or the exam, please do reach out to Alden. You can reach out to me as well. I just may not be as quick about getting back to you as Alden, our embedded tutor, might be. But hopefully that gets you somewhat started on where we are leaving off from now. Okay, so what were we talking about last time? In my recorded lecture, we were talking about viruses, and we had kind of talked about the fact that in this particular chapter, we're going to be looking at 2 different types of viruses. We are going to be looking at animal viruses, ones that can infect animals and us, since we are humans. And then we're also going to be looking at bacteriophages, and those are ones that infect bacteria. So we left off last time talking about their genomes, kind of the differences between the different genomes, but we were also starting to talk about what that means then for kind of the evolution or the way that these strains can change over time. And the slide that we left off on was really talking about if there's a mutation in a viral genome when it's being replicated, and we know that there are more likely to be mutations within viruses than there are in cellular organisms, and that's because they replicate much more quickly and they don't have any type of editing if it's an RNA virus. So with that, I want to jump us back to our lectures. Let's see, I'm going to pull up the lecture we were on, and I believe this was the last slide I left off on in my other recorded lecture. So I'm going to pull this one up a little bit bigger so we can all see it. We're going to talk again about the idea of these viral genomes changing over time. So sometimes what happens, and especially when we grow viruses in the lab, and the way that we grow viruses in the lab is that we grow them within cells, host cells that they can actually replicate in. And often what we see when we grow them in the lab is that they begin to lose their infectivity, so their ability to cause disease, because they're not really kind of in that natural selection. We're growing them in these cells that they can grow in quite easily. And this can often lead to something that we call an attenuated strain. And really that just means that it's weakened. It's unlikely to cause an infection in a healthy individual. Now, when we think about this, the nice thing about this is that these can often be used for vaccines. We can use these. And if you've ever heard of a vaccine that is considered a live attenuated vaccine, that's exactly what it is. It's a weakened virus that we can not infect, but vaccinate somebody with, that will mimic an infection without causing disease. There are limitations to using live attenuated strains for vaccines, in that we don't give these to people who are immunocompromised, because there's a chance it could still cause disease in them. So that's kind of thinking about how they could be weakened. But oftentimes the mutations might be beneficial to the virus. So we're not looking at it from our perspective, right? We're looking at it from the virus's perspective. And some of the ways that it can be beneficial is, first of all, if it changes something on the outside of it. For example, the spikes, it might be that if your body was immune to it, if there's new spikes on the outside, your body might not be able to detect it. And so that would allow it to escape that host immune system. We also see that sometimes a mutation can broaden the host range. And what do we mean by that? So likely with coronavirus, it is very likely that this was an animal virus that did not infect humans until recently. It's most likely that it was actually a virus that infected bats. And what we think is that it likely had some sort of a mutation that allowed it to broaden that host range, meaning not only could it infect bats, but that it could infect humans as well. And we see this quite commonly between viruses that infect animals that can make that jump. So if you remember the swine flu, this was an influenza virus that we saw typically in pigs that made the jump to humans. And that was through a mutation that allowed it to have a larger host range, or a larger number of different types of hosts it could infect. And so that's something that we always are concerned about with viruses. It can also sometimes expand what is called tropism. And tropism refers to the type of tissue or cell that a virus can infect. So sometimes what we see is that the virus is known to infect maybe the respiratory tract of a given virus, and that over time it mutates and can also infect maybe the digestive tract as well. That would mean that it's not changing the host, but it's changing or expanding the type of cells it can infect within one host. And then the last thing that we see that's a beneficial mutation to the virus is one that we saw with Omicron. And in this case, what we saw is that it had an increase of its infectivity. So the mutations in Omicron made those little spike proteins that were sticking out grab onto our cells, or kind of put that key in the lock of our cells more tightly. And so it allowed it to infect much more easily. And so there could be a lot of mutations that are actually beneficial to a virus. So how do these come about, and how do we see kind of this development of new strains in viruses? We're going to talk about a couple of different ways that this can happen. And sometimes what we can have is what is called a reassortment, where we have two different viral strains that might co-infect the same host cell. So let's say that you have viral strain A and viral strain B. They're the same type of virus, but they're different strains of it. And when they infect a cell at the same time, what may happen is when new virus cells are put together or assembled to be released out to spread the virus, is that it may have an assortment of this. And that means that we could have a new genetic combination, which means that virus may have new qualities to it. We actually refer to this, we have a couple of different terms, and I'm going to go to the whiteboard to kind of walk you through what these are. We have what we can call antigenic drift. And then we can have something called antigenic shift. And so you want to think about antigenic drift as being a smaller change. So this one is a smaller change. And this means that the virus, and this is very common I'm going to put that this is very common with influenza. And so what this means is that maybe in 2021, we have particular spikes and markers on the outside of influenza that we vaccinate for. So that year you're protected. You probably know that the next year you are always recommended to get a new influenza vaccine. And that's because it has an RNA genome. And from one year to the next, those spike proteins change slightly. So it still is influenza, but the things that we had immunity to one year, the next year we may not have immunity to. And that's what we call antigenic drift. And this is why something like influenza, we do a yearly shot. It's because it changes enough over the year. It's not a huge change, but it's enough that we're no longer immune. Antigenic shift is something that's a little bit different. And we can think about this of kind of a recombination of two viruses that create a novel new virus. This is a bigger change, so much bigger. And so I had talked a little bit about the swine flu. One of the things that we think may have happened was that pigs were infected with what was typically a pig flu, a swine flu, and they also were co-infected at the same time with the human influenza. And somehow those two viruses, as they were replicating within the cells of a pig, recombined their genetic material into a novel new virus. And this is where we start to see things that can cause a pandemic. Because it's a new virus, there would be no immunity, no natural immunity in the world because it's a brand new virus. So nobody would have been exposed to it already. So I'm going to walk us back to the slide and go over this, but these are ones that you want to make sure that you know the difference of. Antigenic drift is always this much smaller kind of small changes within the one virus, where antigenic shift, we're really seeing kind of a new virus that's emerging through the combination of two others. So when we think about, so we'll talk about the drift first. Following infection or a vaccination for influenza, that host immune system will then be kind of exposed to these spikes, and it will build up an immunity to it. And that is because those spikes serve as what we call an antigen, which our body recognizes and says, hey, we're going to make an antibody that will block this from causing a problem. And so those specific antibodies that we create will protect us as long as influenza has that same spike. If those spikes change, our antibiotics are no longer specific. And so this gives you a good idea of what this looks like. And again, this is antigenic drift. So we can see influenza maybe 1 year, and it has these two different spikes maybe out here, these ones that kind of look squarish and these ones that kind of look round. This person then has a vaccine for it, and they are immune because they have antibodies that recognize these two spikes. Over the course of a year, we have spontaneous mutation, which leads to the fact that these round ones change slightly. And so this is a new flu season the next year. The immunity that this person had that year before will no longer protect them. And that's because influenza has gone through an antigenic drift, meaning that those antigens have drifted enough, they're just changed enough that they're no longer going to be recognized by the person's immune system. And so in this next flu season, we would give another influenza vaccine that would hopefully have this particular spike as one of the targets. That's antigenic drift. It's a much smaller change than we would see with antigenic shift. With antigenic shift, as I said, what we see is often that there is a major genetic reassortment. And as I said, often it is that we have one animal that is basically kind of the host for two different viruses that recombine. So in this case, it's showing a pig lung cell, and maybe they were infected with human influenza and bird influenza or avian influenza. Within the pig's cells, as those new virus particles are put together to be released and actually leave the pig, what we see is that we see this new strain that has some antigens that came from the human influenza and some antigens that came from the avian, and this is a brand new human strain. The problem with this is because it's new, or you've often heard coronavirus referred to as novel, nobody has been exposed to it, which means they have not had a chance to build up an immunity to it, which makes everybody vulnerable. And this is where we see the possibility of a pandemic. And this is one of the main theories of how the coronavirus came about. The thought is that there was likely a bat virus that may have gone through an intermediate host and reassortment that then created this new highly virulent human strain of coronavirus. So that's antigenic shift. Again, that's a much bigger or major genetic reassortment than what we saw with antigenic drift. And those can both be problematic, though. So antigenic shift can lead to that increased infectivity, right? It could cause infection more quickly. Or what we think happened with the coronavirus, it could expand its host range. So making that jump from either the bat or maybe an intermediate animal to humans. And because no person would have any immune protection from an infection or vaccine because it's new, it really did set the perfect stage for the pandemic that we are currently living in. I'm going to pause here. We're going to have a checking in as I've done in the other recorded lecture. What I plan on doing, I'll give you 10 seconds now or so, 10 or 15 seconds to allow you to pause this. If you want to write down the answers before I go over them, you are welcome to do that. Okay, hopefully you paused it, took a second to write down the answers to this. I'm going to go ahead and go over this. And some of this even was from what we covered on the earlier recorded lecture. So the question is, how do viruses differ from eukaryotic and prokaryotic cells? So the biggest difference is they're not cells. So they don't have any of those characteristics that we associate with cells. They're basically just genetic or a genome that is protected by a protein shell. They're much, much smaller, and they must reside inside of a host cell in order to replicate. So they're not considered alive either. They're considered active or inactive. The next question, what are the major structures of a virion? A virion is just one particle, virus particle. And so the major structures of a virion would be that capsid, that protein shell, and then the genome, the genetic material inside. That's the major structures. We could have with animal viruses envelopes, right, which are those membranes that it might have stolen from the host as it exited. We might have spikes, right, those spikes that are kind of the key to unlock the receptors on the host cell. And if it's a bacteriophage, we likely have that complex structure that allows it to attach itself to a bacterial cell and inject the genome into the bacterial cell. So there's a question here asking, what do the genes of a virus code for? I haven't really talked about that, but viruses have very small genomes because they don't have to make up these cellular components. So the genes of the virus generally code for the proteins that are going to be the capsid of that protein and any other thing it might need, like spikes. Sometimes they do bring their own enzymes with them, so they would have the code for those enzymes. The fourth question was asking, what are the different types of genomes found in viruses? So I said that they can be either RNA or DNA. We can also talk about they could either be single-stranded or double-stranded. And then the next question was, which takes the most steps to express a protein? And remember, that's what we covered in the last week. And the ones that would take the most steps are the single-stranded DNA. And then that it would be the, after the single-stranded DNA, we would also see that the one that would take the most steps would be the retroviruses as well, right? And both of those have to make DNA before they can make their mRNA. It also asks which one takes the least amount of steps, and that's going to be the single-stranded RNA positive sense viruses. And SARS-CoV-2 is one that can do that. It can immediately, as soon as it's into the cell, its genome works just as though it's an mRNA, and it can immediately be translated into proteins. The last question asks, what is the process that often causes influenza immunizations to fail? And this really is talking about from year to year. So this year's immunization for influenza would likely fail next year. And the process that makes that is true is antigenic drift, right? That slight change of maybe some of the capsid spike proteins. So that kind of covers the basic kind of understanding that we have of viruses, kind of an introduction to viruses. The next thing that we really want to start talking about is how do viruses replicate? We talked about the fact that they're not cellular, but we want to talk about kind of the steps that have to take place. And we're going to start out with bacteriophages, which are those viruses that can infect bacteria. We're going to go through kind of their replication cycle, and then we will finish up on the replication cycle of animal viruses like SARS-CoV-2. So this looks like a lot of learning outcomes. It is for section 6.3. We're going to talk about the fact that bacteriophage, or viruses that infect bacteria, have two different replication cycles that they could do, and we'll talk about what that means. We'll also talk about the term phage conversion, and this is something that can happen to some bacteria when they are infected with lysogenic bacteria or bacteriophage. And then we'll also talk about the steps in animal virus replication, which is slightly different from the bacteriophage replication cycle. When we get into animal virus replication, we're going to talk about the different ways that enveloped and naked animal viruses both enter the host cell and also exit. And then the last part of this chapter really explains some of what I would consider kind of the long-term effects, or some of the consequences of viral infection. And so we'll talk about the idea that there can be persistent infections, how there can be chronic or latent infections as well of viruses. And then we'll also talk about the fact that some viruses are what we call oncogenic, which means that they can actually cause cancer. And we'll talk about why that is, that viruses have the ability in some cases to cause cancer. So what we want to talk about first is how bacteriophage replicate. And bacteriophage are those bacteria that infect bacterial cells. And I want you to keep in mind that they will infect a host bacterial cell. And their whole thing that they want to do in this lytic replication pathway is basically infect the cell, make tons and tons and tons of copies of themselves, and then burst open that bacteria to go out and infect other bacterial cells. So I'm going to do a quick drawing of this. It'll obviously be a better drawing when we get back to the PowerPoint, but I like to show this. And I want to say that this is talking about bacteriophage. So you can think about that. It's bacteria eating. Phage means to eat, and bacteria. So that's because in this process, the bacterial cells are always going to be lysed or broken up. And we can have two different replication cycles. So I'm going to put replication pathways. Remember, bacteriophage are viruses that infect bacteria. And we have two different, we have one that is called the lytic phase. Oops, not phage, but phase. And then we can have something called the lysogenic phase. And so I'm going to walk you through kind of the steps of each of these. Remember, these are the replication pathways. It can do one or the other of this. Organize this a little bit better. And the lytic phase is going to go through these 5 steps. The first step is what we would call attachment. And sometimes you'll see it as, sometimes some books call it adsorption. And you want to think about that bacterial cell, right, that has its chromosome in there. And this is the point where that really cool kind of spaceship-like bacteriophage can land on it. Okay, that's that attachment stage. The second step that we would get would be penetration. And that just means that that genome gets injected, the genetic material gets injected into the bacteria. Sometimes you'll hear this referred to as entry. I like penetration better because that's really what's happening. The third step then is what we would call synthesis, or sometimes you'll see it referred to as replication. And this is where basically that genetic information that was injected in, which I'll show up here, is like an instruction manual. And basically what it does is it hijacks that bacterial cell and it tells that bacterial cell to start making lots of capsomeres that will make up new capsids. It'll make it make lots of copies of the DNA or RNA, depending on what kind of virus this is. And so what you'll end up, oops, let me do a different color. Keep this in mind. Let me erase that. Sorry, I don't know how I got that up there. Get rid of that. I have some sort of a ruler now. I'm not sure how to get rid of that. Oh, maybe I can just slide it off. Sorry. It's the first time that's ever happened. So what we can think about here is if this is that bacterial cell down here, right? And up off of it, it still has the shell of the bacteriophage, but it doesn't have the genome because that got injected in. What we would expect to see then inside of the cell is a whole bunch of these parts being made. So genome, capsomeres, all of this would be synthesized. And that brings us to our fourth step. And our fourth step is what we call assembly. Sometimes you'll see it referred to as maturation. But the really cool thing is that in assembly, these virions, these little spaceship-like things, will just self-assemble. They'll all come together. So the capsomeres will come together around genomes. And what you'll end up with is basically you'll end up with then this bacteria now that is filled with a bunch of bacteriophages. Right? It'll have thousands of these or tens of thousands, and they each have their own little genome in them as well. And that just self-assembles. And then we have our last step of this, which is called release, which means that all of these guys are just going to burst out of the cell. This will destroy the bacterial cell. And often you'll see that as lysis. Okay. One of the things that I want you to keep in mind is in the lytic phase, the word lytic is shorter than the word lysogenic. And so I'm going to say that this is the short replication cycle. The lysogenic, which is a longer word, this is the longer, longer. Let me try writing that a little bit lower. Not sure how I get rid of my, oh, I got rid of it. Never mind. This is then what we can call a longer replication cycle. And so in this case, with this one, we do the same first step. We do attachment or adsorption. We do the second step as well. We do penetration, sometimes called entry. And then we have a new third step. And so I'm gonna color this one a little bit different because this one's a little bit different. We have a third step that's new. And this is where, in this case, the viral genome inserts into host chromosome. So basically the viral genome, when it enters the bacterial cell, rather than directing the cell to start making viruses, it actually puts itself into the genome of that bacterial cell and it hangs out there. And that bacterial cell is likely going to reproduce often and fairly quickly, right? Because bacterial cells can reproduce every 15 to 20 minutes. And all of the new bacterial cells that then would be produced in reproduction would carry this viral genome. So we might start out with a bacterial cell here that has its own genome, but it ended up with a little bit of the viral genome. When it goes on and goes through binary fission, that is going to be copied with its chromosome into those two new daughter cells. And when those two new daughter cells replicate, it's going to be carried into them. And so it kind of can hide out here and just allow itself to be replicated within each of the new bacterial cells. And at some point, maybe the bacterial cell gets stressed, maybe the environment is not so good. It is then going to go back into what was kind of that lytic cycle, and it's going to do synthesis. It's going to excise itself, so it'll leave that chromosome and it'll start to do that synthesis or replication. And this will happen in all of the cells that it is now in. It will then do the assembly or maturation. And lastly, it will do release. So the lysogenic phase has an extra step. And when that viral genome puts itself into the host chromosome, at this point, we call it a prophage. So before being a phage. So it kind of hangs out there. I think of it as being kind of dormant or hibernating until a time comes out where it can start to make those. But hopefully you see that we had 5 steps in the lytic phase, and we have 6 steps in the lysogenic phase. And this is really what happens with bacteriophage in bacteria. Now, there is something that happens like this in animal cells for some viruses. So for example, HIV is one that can actually become what is called a provirus and hang out in our chromosomes before really making a lot of viruses. And we'll talk about that at the end of the chapter. But this is really kind of just showing you the 2 different ways. And so really what you're looking at is that lysogenic phase just adds a step. I'm going to bring us back now to the PowerPoint because it has a much better drawing than I drew, but hopefully the same thing for me. It's helpful when you draw these out. So these are the 5 steps of that generalized bacteriophage replication. We have the attachment, where it actually binds to the bacterial cell. We have penetration, and that's when it injects its genetic material into the host cell, into the bacterial cell. We then have replication, right? That's where it takes over the host cell and it begins to transcribe and translate all those viral genes into the proteins that it needs. It makes copies of its genome. And then we go into step 4, which is assembly. Those genomes get packaged into those capsids and phage structures. And so we get assembly of all these virions. And then the last step is that cell, that bacterial cell, will burst open and all these viruses will escape out to infect new cells. This is a great little diagram of it, a little bit better picture than mine. So you see the bacteriophage, the virus right here. It has attached itself to a bacterial cell that has a chromosome. It will insert its genome into here, and that genome then will begin to direct the cell into making new capsids, new genomes, new parts that would need to be for a new virion. We then get assembly, these self-assemble into new virions, and the last step is release, where they burst out of that bacterial cell, destroying it and going on to infect new ones. If you will, it's a little bit like an alien, right? This little thing kind of landing on a bacteria and then making lots and lots of copies of itself to go out and infect other cells. So that's the lytic cycle. And again, this is the short replication cycle. It just has 5 steps. It gets in, produces all the parts it needs, gets those assembled, and then it leaves and goes out and to infect others. But we can also have what I said was lysogenic replication, which is a slower, because there is a kind of a hibernation stage in the middle of this, or step. And so this lysogenic replication can be done by what are called temperate phages. Some phages have the ability to do this. And so in this case, we have attachment. That's the first step. The second step then is penetration. And then this third step is that added one. Remember, that's where we have that phage becomes incorporated into the host cell genome, and we call it a prophage. As that cell divides, it gets copied along with that prophage, so it continues into all those other cells. And then what we end up doing is that host cell, at some point, if it's stressed, that prophage will get itself out of the chromosome and go back into the lytic pathway at that synthesis step. So it kind of takes this little, I shouldn't say little, it's kind of a long route instead of a shortcut. It's a long cut, if you will. And this is kind of adding this to what was our lytic. So this is what we looked at earlier. This is the lytic, just this whole left-hand column of it attaches, penetrates, it does synthesis, assembly, release. But if we have our lysogenic, after it penetrates, it does that integration into the chromosome as a prophage. This goes through cell division. And at some point, it's going to bring itself back out, and then it will jump back in and start doing exactly where it was in the lytic phase, begin to replicate all the parts it needs, assemble them together, and release. So lysogenic, remember, is the longer word. That means it takes a longer pathway to get to the end result. But this is really what happens with bacteriophage. If this is confusing to you, please do reach out to Alden on this or draw this out. But this is, in both of these cases, the end result is that we have bacterial cell that is releasing lots of virus, but we would expect to see more bacterial cells that have that genetic information and therefore can release the virion in the lysogenic replication cycle. So we're going to shift gears from that bacteriophage, and we're going to start thinking about, instead of bacteriophage, thinking about animal viruses. And we'll talk about that in just a second. So one of the things I wanted to let you know, and I'll go back one slide and talk about this, is many scientists have started to think, because we have this problem with antibiotic resistance, that maybe one of the ways to fight bacteria that cause infections in us is to use bacteriophage against them. Bacteriophage cannot infect our cells. They're specific for bacterial cells. So the idea is, let's say you have a horrible antibiotic resistant bacterial infection. Could we treat you with a bacteriophage or a virus that would attack the bacteria for you? So kind of the enemy of your enemy is your friend. So there's been a lot of talk about doing what is called phage therapy, that we would treat people with bacteriophage that would wipe out the bacterial infection. But one of the things that has become kind of a complication as we've started to study it is the idea that sometimes when these viruses are prophages in bacterial cells, so when they've inserted themselves into the genome, they can make that bacterial cell more pathogenic. And we've seen this with two bacterial species that we really don't want to make more dangerous than they already are. So with Corneobacterium diphtheriae, which causes diphtheria, it's a horrible infection, or Clostridium botulinum, which causes botulism. In both of these cases, when these bacteria, remember these are bacteria, when these bacteria are infected with a bacteriophage or with a virus, they actually become more damaging to us. They cause more disease. And so this has kind of been one of those places that we're looking to see if phage therapy is really going to be a good way out of antibiotic resistance. So we're going to jump now ahead to animal virus replication. We were talking about those viruses that infect bacteria. Now we're going to look at the ones that can infect us. An animal virus replication pathway has 6 steps. And you might be curious why it has one more really than the lytic phase. And it's because of the fact there is a big difference between bacteriophage versus animal viruses, in that bacteriophage, the only thing that enters the bacteria is the genome. But in animal viruses, the whole virus particle will enter. So I'm going to go to the whiteboard and draw this out, and then we'll look at it on the slides. So instead of bacteriophage now, we're going to be looking at animal virus, the replication cycle. And in this case, we have 6 steps. I'm going to write these down. So the first one is attachment. And this is often through the use of spikes. I'm going to put often using, using spikes, right? So they can attach to our cells. And then the second step is going to be penetration. So far this looks fairly similar, right? They're going to enter the host cell. But the really interesting thing is this is the whole virion, meaning capsid and all. And so we have to go through this step that we didn't see with bacteriophage that is called uncoating. And this is the removal of the capsid. And then it looks just like what we saw with the bacteriophage. Then we have our synthesis, right? where we make all of the parts for new viruses. We're going to have that assembly again. And then the last, which can be a little bit different than the bacteriophage, is the release. And so for this, enveloped viruses will, what is called, bud off from host cell. And this allows them to pick up some of the cell membrane with them that will kind of cloak them. But naked viruses, those that don't have the membrane, will burst out of cell. And we'll look at how this looks. They look a little bit different. The naked viruses generally destroy the host cell, whereas the envelope viruses might not ruin it, may not burst out of it because they're just budding out with taking a little bit of that cell membrane with them. So I'm going to walk us back and look at this in the slides, kind of go over these. Get back to our... And so again, these are the steps: attachment, penetration, uncoding, then that replication where the cell is kind of a factory to make viruses, assembly. So these virions, again, just self-assemble. The capsids form around the genomes until they're all kind of made, and then they're released from the cell. This is showing a really nice overview of that animal virus replication. It's quite simple, remember, because these are very simple microbes. In this case, this one is actually an enveloped virus. I can tell that because it has the capsid as kind of this aqua blue structure. And just outside of it is this gold membrane. This is actually an envelope that was taken from the last host cell it was in. And so when this comes in, this membrane will blend in with the new host cell's membrane. So we have that first one, we have attachment, and you can see here the spikes are attaching to some sort of a receptor on the host cell membrane. When it attaches, the host cell thinks, hey, I should let this guy in. And so it allows the virus to come in. The envelope will merge with the cell membrane, and the capsid is what is brought in with the genome. That's that second step of penetration. The third step then is what we call uncoding. And this is where enzymes within the cell will break down the capsid that was surrounding that genome. And that's the end of uncoding. And then we go into replication. This is where capsid proteins are made, copies of our genome are made for the virus, and then spike proteins are made. So all of the things that are needed to make new virions will happen in step 4. We move on to step 5, and this is where we get these just self-assemble. So the capsids form around the genome, and the spikes get ready to be embedded into that membrane as these ones bud off. Remember, these ones will bud off because they are enveloped viruses, and so they're going to take part of the cell membrane with them. If this was a naked virus, the spikes would attach themselves to the capsid, and these cells then would burst out, basically breaking open the host cell. And so this will go through a little bit more step by step and showing the difference between the naked versus the enveloped virus. In this case, it's showing that sometimes the capsomeres can act as what attaches to a host cell, although naked viruses can also have spikes. So they could also attach through a spike. But we see attachment in both these cases to the host cell. The second thing that happens is penetration. And as I said, this looks slightly different. If you're enveloped, that membrane, that gold membrane around it will merge with the host membrane and just release the capsid inside. If you're a naked envelope, or excuse me, a naked virion, you'll be brought in to the host cell. And then again, you will have that capsomere broken down. Then we have the uncoding, and this is where I said that capsid, that protein shell that's around the genome, will be broken down by some enzymes. We have replication, step 4, where the genome is replicated and all those viral proteins that are needed are made. And then lastly, we have assembly. All those new virions just self-assemble. Now, depending on if you're naked or you're a enveloped virus, you may leave the cell differently. And so this is showing one that is enveloped. It is already self-assembled into its capsid. And as the capsid is going to leave the cell, it picks up some of the host cell membrane that has the spikes embedded in it, and it buds off. And then we have our enveloped virus. And this is a picture of HIV, which is an enveloped virus, doing just that. You can see that it's taking some of the cell membrane with it as it leaves the cell. If this was a naked virus, it would just burst out of the cell membrane and likely cause the cell to rupture. So let me go back. That was the end. So I just want to pause there. That was the end of our animal virus replication. Hopefully you can kind of look at these two and compare both the naked and enveloped animal viruses, but also it's important to look at the difference between the animal viruses versus the bacteriophage. And the biggest difference is that with animal viruses, the entire capsid and genome enter the cell, and in bacteriophages, just the genome enters because it can inject that in. So that is the end of our two types of viruses, their replication cycles, and we're just going to talk a little bit more about animal viruses and kind of the way that we see them on how they can cause infection depending on the type of virus. And then we'll also talk about oncoviruses before we end this chapter. So animal viruses, because of the way that they replicate, some of them can cause different types of infections. So probably the one that most of us are familiar are what we would call an acute non-persistent infection. This is something that you would see with something, say, the common cold, measles, influenza. And what would happen is that you would have this big increase in viral replication, you would get quite sick, and then your body would fight it off and it would go away and you would be done with that infection. And that's quite common with a common cold. That's considered acute non-persistent. It means that you don't have symptoms or infection from that virus again in the future. But we can also get ones that kind of stick around, and they can be either what are called latent or they can be chronic infections. So a latent persistent infection is one that maybe you have a outbreak of the particular virus, and then it goes down and we don't really see any symptoms or signs for a period of time that can be days, months, or even years. And then maybe you have another outbreak and then back down, and then maybe another outbreak. The really common one that we see with this falls into either the herpes virus family or chickenpox, right? So we know that with herpes, even ones that we see like cold sores around the mouth, we can have these outbreaks and then it kind of goes dormant for periods of time, and then outbreaks, usually related to stress or fatigue. We also see this though with chickenpox, right? You might have had an initial chickenpox infection, and then years down the road, you might end up having shingles. And so these are what we call latent persistent. The body never clears itself of the actual infection, but we go through long periods of time where we don't actually have signs or symptoms from it. The last type of persistent infection that we're going to talk about is what is called a chronic infection. And in this case, we start out with very low levels of viral replication, and these begin to increase over time. And that can be days, months, possibly even years. One that we would imagine that would fall into this category would be HIV, that many people could go months and even years before they start having signs or symptoms. So we're going to talk about each of those persistent infections a little bit more. So as I said, those latent persistent infections are those ones that we often have these flare-ups, and then we have these intermediate periods of no signs or symptoms. It's during those flare-ups when you're likely to be infectious, so you're shedding these virions, and you would be experiencing symptoms. These can be triggered by stress, often by fever, or sunburn can sometimes do it, hormonal levels. Those changes can also flare it up. So we can kind of think about that. Again, we think of herpes and chickenpox. And so any of the members of the herpes family are notorious for causing these latent persistent infections. So herpes virus 1, which is cold sores that we find around the mouth, herpes virus 2, which we found as genital herpes. This is one that we see as a sexually transmitted disease. And then lastly, we can see that varicella zoster, which is the herpes virus that causes chickenpox and ultimately can cause shingles down the road. These are ones that we know kind of follow that pattern of you have a flare-up, and then you have this latent period, and then a flare-up, and then latent period. So we call those latent persistent. The other one that we want to think about are those chronic persistent. These are characterized by this kind of continuous release of virions over very long times, usually months or years. And we have a very slow progression to disease. HIV is a perfect example of this. Many times people don't even have signs or symptoms for months or even years. And then we kind of see it slowly progress over time. And I do want to show, because HIV and chronic persistent infections are similar to that lysogenic pathway that we saw with bacteriophage, and that they kind of hibernate within the cell, if you will. And they have an added step to what we think of as the typical animal virus replication cycle. So again, we start out with attachment and penetration. We do uncoating. And then in this case, what they do is they do this reverse transcription. So we have this viral RNA genome. And HIV can carry a enzyme with it called reverse transcriptase, which allows it to take its RNA genome and reverse transcribe it into DNA. That DNA then can be inserted into the host genome, into the chromosome, and hang out there for a period of time. We call it a provirus at this time, which is similar to the prophage that we saw in bacteriophage. And then at some point, it will excise itself from that and go through the synthesis, assembly, and release. So that's one that we see with chronic persistent infections. And as I said, HIV is a very good example of this. The last part of this chapter on viruses, I want to talk a little bit about the fact that we can have persistent infections that actually can lead to cancer. They have the ability to actually cause cancer. So we have viruses that we can either refer to them as oncogenic, so onco meaning cancer and genic meaning genesis or the birth of cancer. They can be shortened also to oncoviruses. And these are viruses that we know can cause cancer. About 10 to 15% of all cancers, we have been able to find a viral cause to. And what they can do is they cause cancer by stimulating that host cell to begin to do cell division and not stop. So do uncontrolled cell division, or to decrease that host cell, its ability to respond to when it should not be continuing to live. It should be going through death, that it would continue to divide. Some examples of this, probably one of the best known ones is human papillomavirus, which causes, it can cause cancer of the cervix in women. It can cause penile cancer in men. It has been proven that it actually can cause cancer if it goes untreated. We also have some human T lymphotrophic viruses that do very much the same thing. They can cause cancer through infection if not treated. So these are some of the issues besides just an infection that this virus can do. It can also cause these long-term consequences of cancer. So I'm going to pause here. Again, I'll give you about 15 seconds to pause your video of this so that you can write these down. And then I'll come back and I will answer these questions. So question number one, let's define bacteriophage. Remember, bacteria refers to bacteria and phage means to eat, or phage, to eat. So these are viruses that can infect bacterial cells. The second question is, what is the difference between the lysogenic and the lytic phase? Remember, the lytic phase is the short word. That means it's the shorter cycle. So that means that the virus gets its genome into the bacterial cell, it replicates, it assembles, and it releases. The lysogenic is the longer word. It means it's the longer cycle. So after attachment and penetration, it will integrate its genome into the host chromosome until eventually it can excise itself and return into that synthesis, assembly, and release stage. So it has an extra step, the lysogenic does, and that's the integration into the host cell genome. The third question, what step do animal viruses go through that bacteriophage do not? That's the uncoating step. And that's because the capsid of animal viruses enters the host cell, and bacteriophage, the capsid does not enter the host cell, only the genome does. Question number 4, name a chronic infection. So a chronic infection If we're talking about a chronic one, HIV is a good example of a chronic infection that we see kind of increase over time. A latent infection, remember, that's one that has periods of kind of latency and then periods of flare-ups. And these would be any of the herpes virus or chickenpox virus. And then the last question, what does oncogenic mean? Oncogenic refers to a virus that can cause cancer. So that completes all of our stuff on viruses. If you have questions on any of this, please do reach out to Alden. If you want, you are welcome to email me. Just remember, I may not be able to get back to your questions as fast. The last part of this chapter, we're going to talk about something that's really interesting and is a little bit near and dear to my heart, and I'll explain why that is. We're going to start out talking about the different drug approaches that can be used to manage viral infections. We'll talk about some of these antiviral drugs. You do not have to memorize the antiviral drugs. Basically, you just have to know what the targets are for antiviral drugs. And hopefully, you know, we don't have a ton of antiviral drugs, but we do have some. And then at the end of this chapter, I'm going to talk briefly about prions, describing what a prion is. If you've heard about the wasting disease that has been found in elk and deer in Idaho, this is caused by prions. I'll explain what that is. I'll also talk about a disease that they can cause in humans that you might have heard of and explain how it can be transmitted. So moving forward. So antiviral drugs can treat infections, but they can't cure them. And one of the things that we want to think about viruses is the fact that there are some real obstacles into targeting viruses. So the way that we target viruses is we look at that viral replication pathway. So if you go back and you review the animal virus replication cycle, we target those 6 steps. So we target either attachment, penetration, uncoating, synthesis, assembly, or release. And that's because there are not great targets in a virus. This isn't a cell that we can target some cellular structure in them. And so one of the things that we want to think about when we design these antiviral drugs is, first of all, the fact that viruses are obligate intracellular pathogens, which means their replication cycle takes place inside of the host cell. So it's very hard to target them if they are inside of our cells without harming us. Remember, the goal of anything that's an antimicrobial is to be what is called selectively toxic, that it kills the virus or inactivates the virus, but doesn't harm the host. And that's hard if they're inside of our cells. And because of that, it's very difficult to come up with antivirals. They also have fewer chemically distinct targets than living pathogens. Like they don't have ribosomes that we can target, or they don't have a cell membrane or a cell wall to target. So that makes them difficult. So the best kind of, not treatment, but the best way to really rid ourselves or prevent serious viral diseases is through vaccination. That's the safest and easiest way to protect ourselves. Vaccines actually will train the immune system to recognize the virus so that if you are infected with it, it fights it off before you get sick. And we'll spend more time on this. I believe it's in chapter 14 that we do a lot on vaccines. So this is a lot of information on the slide. Again, you do not have to memorize these drugs by any means or what virus they target. This is just to give you an example. But remember that drugs will target each of those steps that we might have in the viral replication cycle. And so they could block attachment, making sure that something doesn't attach to a host cell. They might block penetration. So even if it's attached, they stop that capsid from entering the host cell. Some drugs can block the uncoating process so that it can't get the capsid off, so that genome is not able to direct anything. A common place that we see it is at replication and assembly. Even one of the drugs for SARS-CoV-2 is looking at blocking the RNA replication, so the genome of the coronavirus. We have one that tries to block that so it can't make more copies of its genome to put into new virus particles. And then there are some that try to block the release, basically the release of those viruses from the host cell. So the good news about learning how antivirals work is that all you have to do is memorize the steps of the animal virus replication cycle, because that is the targets. So this is a good chance to go back and look over your animal virus replication cycle. As long as you know those 6 steps, you could say that's one of the places that an antiviral would block replication. All right, on to the very last thing in this chapter, I believe. And this is talking about prions. So interestingly enough, my own little personal experience. So I like to usually, this is kind of my chance to tell you if anybody has ever said that I'm kind of crazy or weird, I blame it on the fact that I could be at risk of having a prion infection. And I'll explain kind of to you what that is. As a matter of fact, both of my mother and my stepfather both also are people who were at risk of having a prion infection. Which means we're not allowed to give blood in the United States. And I'll talk a little bit about what it is that could have been contaminated. So prions are really interesting. They're not cells. So remember, they're outside of that kind of concept map that I drew the first part of the lecture, my first pre-recorded lecture. They're just infectious proteins. They're not viruses either. All they are is a protein. But they're infectious proteins, and they can't replicate. The thing that they do is that if you're infected with this type of a protein or prion, is that these prions, when they get around your other proteins that are normally in your body, they cause those proteins to fold improperly. And one of the most common ones that people know about, or that has been studied, is one that's called transmissible spongiform encephalopathies. And these are ones that do damage to the brain. They basically turn the brain, so this is a normal brain tissue, and they turn it into a sponge by putting these holes in it. Basically, because of the misfolding of the proteins in the brain, you start to have these big spaces in the brain that are no longer functioning. So if you've ever heard about mad cow disease, that's exactly what this is. And the problem was with mad cow disease is that these cows that were infected with the prion, they were often, they looked injured. They couldn't physically stand very well. They were definitely these ill animals. And what happened in the I want to say it was the 1980s and early 90s, was that back then, they did this horrible thing of when animals died, or they put animals down like cows, is that they would often grind them up and add them to the feed, because that made the other cows bigger. But it also spread this particular type of acellular non-viral infectious agent. And so mad cow disease was actually quite common in the UK and the United Kingdom at this time. There were outbreaks of it. And I happened to live in the UK during that time, along with my parents, for longer than 3 months. And since we lived there for longer than 3 months, we're considered at risk. Now, this was 30 some years ago, so I don't think we really do have it. I think by now it would have presented itself, but it is one of those things that we don't have a cure for. And we actually don't know if somebody's been infected with it or has it until they do a biopsy of the brain. So the ones to think about, the different types of spongiform. So the one that's found in humans, the one that I would have been at risk for, which I don't think I am anymore, because I think I'm long enough out from it, is called the Creutzfeldt-Jakob disease. And this is basically caused by the same prion that causes bovine spongiform encephalopathy in cows, or otherwise known as mad cow disease. Lucky for me, I didn't eat a lot of beef when I was in England, but my mom and I always do joke that my stepdad ate things like kidney pie. So if anybody's going to get it, it's going to be him. There's another one called Gertzmann-Straussler-Schenker syndrome. It's very similar. But this is one, if you've been reading the news, or if you're somebody who hunts, has been has been identified in many elk and deer in Idaho this year. There have been, I think, 2 to 4 elk and deer that they have identified it in. So there's this big push that you should make sure to get the meat checked before you were to consume it. It's a really weird, the fact that it's just a protein. It's one of the highest biosafety standards that we do to study this particular type of disease. It's actually at Rocky Mountain Laboratories in Montana. They do a lot of research on prions there, but there is a lot understood, but there's also still a lot to be learned about it. So Prions are not viruses. They are not cells. They are infectious proteins that can be quite devastating to people or animals that are infected with them. So with that, I'm going to pause here and give you guys again another 15 seconds or so to pause it. And then when you come back, we'll go ahead and go through these. All right, so hopefully you paused it, wrote down the answers. So in the checking in, I have 3 big questions. The first one is, why are viral infections difficult to treat? And there's a couple different reasons. Number one is there aren't a lot of great targets, right? The virus is only a genome and a protein shell. There aren't any structures besides that, like a cell wall or a cell membrane or ribosomes to target. It's just that. The other thing that makes them difficult to treat is that they replicate inside of a host cell, and it's very difficult to get to them once they're inside the host cell. The second question asks you to name at least 2 targets that antivirals work against. Remember, the targets are just the steps in the animal virus replication cycle. So you could target attachment, penetration, uncoating, synthesis, assembly, or release. Those are the 6 that you could target. And then the last question was, name a non-cellular. So if it's non-cellular, we know it's not bacteria, it's not fungi, it's not protists. So it's non-cellular, non-viral, it's not a virus, infectious agent. There's only one that would fit that category, and that would be a prion. So yeah, if I hear any funny jokes about me, just remember it's just the prion, it's not me. So that should be the end of chapter 6. I do want to jump to the whiteboard and remind you that there should be, you have the ability to go in and do the Kahoots for chapter 6. All you have to do is go to kahoot.com. You can look under my name, all one word, all lowercase, no space, of Holly Paquette. And there should be both the chapter 5, so on genetics, and the chapter 6. So you can do chapter 5 and chapter 6 should be available to you now. Okay. That is the end, at least for us, of chapter 6. I have about 10 minutes, I think, where I'm at least going to get us started on chapter 7. So I'm going to jump us into chapter 7, get us started a little bit in there, and then hopefully the next time that you guys see me will be in person, not some recording of this. So let's go back, and I'm going to bring up chapter 7 and get us started. And we're going to be talking about microbial growth in this chapter. And we're also going to be talking about microbial control. This chapter for me, I will say is I don't want to say boring, it's not the right word, but it's very vocabulary driven. And there aren't kind of the processes of like DNA replication or protein synthesis or viral replication. A lot of this is memorization. And that's why you have 2 sets of vocabulary in Sarago. There's a lot of vocabulary. So please take the time to really look over the vocabulary for this. And hopefully at least I can get a start on this. You guys will feel like you have a good start on this chapter. So we're first going to review binary fission. We've talked about that a little bit, about one bacterial cell can split into 2 new ones. That's what it does through cell division, and that's how we get more bacteria. We're going to talk about how you can calculate the generation time for a bacterium. So how long does it take from 1 cell to go to 2 cells? How long does it take from those 2 cells to go to 4 cells, and so on. Oops, let's see if it's going to let me back. Oh, hopefully I'm still... Let's see, return and see if we've got it. Not sure what happened there. PowerPoint ran into a problem while I was doing it. Let's see. So I'm going to cancel that. Okay. So hopefully it's going to let me do it. Okay. We're going to bring that back up into our slideshow. And so after we do those generation times, we'll also talk about the 4 stages of bacterial growth and what is called a closed batch system. So if you were to just grow bacterium in the lab by itself in a broth, what it would look like, how its growth curve would look. And then we'll talk about how that looks different, though, when we think about going into nature where there's other things that are competing with that bacteria. So with that, we want to think about microbes and their ability to grow in nature, and then how they might grow differently in the lab. So in the lab, we generally grow bacteria as a pure single species culture. So they're not mixed with other bacterium or other things that they might be mixed with in nature. Because we know in nature, bacteria will be mingling with archaea and eukaryotes and viruses, everything. But in the lab, we try to just work specifically with those pure cultures. We know that environmental factors, though, can play a significant role, right, in how healthy that bacterium is, what kind of nutrients are available for it to metabolize, and even the structure of the bacteria. We also know that they can form biofilms that can protect them in surviving, but also help them communicate in their environment. We know that those biofilms, though, can be a major concern when they are in the healthcare setting. They are difficult to treat, and they also can contribute to persistent infections. With that, we want to talk about how bacterial cells divide. And you guys already have gone over this in earlier chapters, but we're going to talk about it again because we're going to talk about the idea of their growth curve. So in that process of binary fission, that cell, before it goes through cell division, is going to do DNA replication. Remember, that's when DNA replication always takes place, is just prior to cell division. And so the first thing that will happen is before dividing, the chromosome is replicated, and that's DNA replication that we covered in chapter 5. Then each of those 2 chromosomes, because now there are 2, one will move to one end of the cell and one will move to the other end of the parent cell. And then there begins to pinch off. And you can see that happening here, where it begins to put a partition between the 2 halves of the parent cell. Those will eventually completely separate, and we will create 2 genetically identical daughter cells. And we think of that when it goes from 1 cell to 2 cells as a generation time. It's the amount of time it takes for 1 cell to divide into 2 cells. And this is dependent on a lot of things, right? It can be dependent on how much nutrients are available to that microbial population. It can depend on the species and the conditions in which they live. Bacteria, though, usually have a replication or generation time between 15 minutes up to 24 hours. For many common bacteria, the generation time is actually less than 1 hour. So like E. coli, which might be in your potato salad, the generation time is 20 minutes. It's quite quick. So 1 E. coli can become 2 in 20 minutes. Another 20 minutes later, you could have 4. Another 20 minutes later, you can have 8. So it's this exponential growth. There are some bacteria that have fairly slow generation times. The bacteria that causes tuberculosis, Mycobacterium tuberculosis, is usually in the hours, like 15 to 20 hours to get that. But we want to think about how we can figure out the generation time. And so the way that we look at this is we can look at, if we were to grow a particular bacteria for 1 hour, we could actually look at how many generations we see of growth, and then we can backtrack, do the math to figure out what the generation time is. So if we have 1, 2, 3 generations that happen in that 1 hour, that means we can divide 60 minutes, the total time, by the number of generations, and that tells us how long each generation is. It's 20 minutes between each of them. So that's one of those things that we think about as those bacteria divide through binary fission, and we get that exponential growth. I'm just going to briefly introduce this, but this is likely the slide that we will come back to when I am back in person. But bacteria have 4 distinct growth phases that we would see when we grow them in the lab in kind of this, what is called a closed pure batch system. This means that it's just one bacterial species in a flask full of broth. And what we can do here is we can let it grow. And what we see along the y-axis, so going up here, is the number of cells in the sample. So as it goes up this way, there's more cells that are viable or living cells. And then across the x-axis is time. So over time, what happens? And we can see these 4 distinct phases. We have what is called the lag phase. And this is where we don't see an increase in population of the bacterial cells. You can see it's a flat line. And that's because the cells are adjusting to this new environment of being put in the broth and getting ready to do cell division. The next phase that we have is what is called the log phase. You'll sometimes see this called the exponential phase. This is where we have exponential growth. The bacterial cells are just doubling they're having a new generation over and over again very quickly. And so we see this incredible increase of the number of cells over time until we reach a point where we hit what we call the stationary phase. And again, we get this flattening where we don't have an increase of cells or a decrease. And that's because the number of new cells that have been formed through division, cell division, is equal to the number of cells that are dying, because we're starting to get crowded in this space. There's waste that has been given off by some of these bacterial cells that is toxic to other cells. And so what we see is we don't see any growth anymore. And then the last one that we go into is what is called the death phase. And at this point, more cells are dying than cells that are being created. And that's because there aren't very many nutrients left in the broth. And there is lots of buildup of waste and dead cells that can actually be toxic. And so this is the one that we'll come back to. But this is really the growth curve, that standard growth phases that we would see for bacterial cells. So I'm going to pause it here. This is the end of the second lecture then that should have been for 3-3 for March 3rd on Thursday. And then hopefully what I hope is to see you guys all back the following week. So please do send out any questions that you have to Alden is probably your first choice. I am probably your second choice because I don't know how fast I will be at getting back to you. I hope you have a terrific week, and I will plan on seeing you on the 8th of March. Take care, and I will see you soon.